9th, 2020. We're working on our second to the last unit, medical microbiology, and this is part 2 of medical microbiology. The first one was on virulence factors, and the second one is on epidemiology, disease ecology, reservoirs, and transmission. So it's a long one. I added a lot of new stuff for the COVID pandemic coronavirus. So I'll break it up into 2 videos. Okay, so let's see here. All right. And again, folks, I added a lot of links, and a lot of the slides are full of text from articles that I thought was of interest with regard to the COVID, but you won't have to look at all those links nor memorize all the text, okay? I'll try to, in addition to the study guide that's posted on Canvas, I'll also try to do some more practice quizzes. And basically with the practice quizzes, I'm just taking your study guide questions and turning them into quiz questions, okay? So we are really interested in infectious disease. So if we just kind of tear that apart, so disease, generally you could say it's interference with any normal bodily function. So there's an upset in normal homeostasis, and there can be many different causes. We know that cancer can be a disease. So when we're talking about infectious diseases, we're talking about a microbe invading another organism and causing harm. So specifically with infectious disease, and I really should put that in here, that it should be, let's do that. Let's put infectious disease in here so we're a little bit more specific. So if we're talking about infectious disease ecology, there's 3 factors that contribute to the occurrence of infectious disease. And we'll see in the next slide, often we'll use a Venn diagram showing how host factors, environmental factors, and microbial factors, they have to be just right. Just when the factors are just perfect, kind of like the perfect storm, that's when we'll have infectious disease. So with regard to the host, you consider the age, the immune status, their nutritional status, the level of stress they're under, if they've had any injuries or surgeries. All of those factors can influence whether infectious disease can occur. With regard to the environment, we think about climate, we think about, are there arthropod vectors around, are there reservoirs around, what about the cleanliness of drinking water, the cleanliness of food, for example, and then the microbe itself. So we want to remember that most microbes don't cause harm, right? It's really an unusual property to cause harm, right? And in the previous PowerPoint, we talked about virulence factors. So some microbes are loaded with virulent factors, right? So we could argue that most times if they infect us, we're going to have disease. But we also want to remember that our normal microbiome, the normal microbiota, that it normally, if it's living where it's supposed to live, and if our immune status is good, our normal microbiota should act as commensals and even mutualists. But we do know that some of them, like for example the yeast Candida albicans, if it's given the opportunity, it can switch from being a commensal to becoming a parasite causing harm, right? So we always need to think about the normal biota, the normal microbiome, and if given the opportunity, if it perhaps could cause harm. So this next slide again, folks, is just this Venn diagram. And again, just stressing in the ecology of infectious diseases, host factors have to be just right, microbe factors just right, environmental factors just right. So it's where these factors intersect, right? That's when we'll have infectious disease. And this is traditionally, I usually start introducing you to this concept of the One Health Initiative, an ecological approach to human, animal, and environmental health at the start of this semester. And because we started with our new microbiology textbook, OpenStax, I left this one out. But here at the very end, I think this is very fitting. We do want to remember that for us to maintain the health of humans, animals, and the planet, we have to take care of all, all populations, all of our environment. We have to take care of it because if one part, again, kind of see this almost like a Venn diagram here, you guys, if any part of this becomes sick, right, then we would argue that the health potentially of all the organisms on the earth could be impacted. So again, folks, we want to, if we're talking about infectious disease, we're really talking about a type of symbiosis. So we want to just go over symbiosis again. So symbiosis is when we have two or more different organisms living together. And there was one report when they started doing the genetics of the human intestinal microbiome, and there was one report that said that for every human cell there were 10 microbes, right? And so people were saying, so are we more microbial than we are human, right? And then the next question would be, well, if there's so many microbes present, why aren't we always sick, right? So we can see that microbes can have different relationships living with us. So a lot of times they're actually helpful. They're part of our natural defenses, right? So we just want to go through these three types of symbiotic relationships. So mutualism is when the two symbiotic partners, one and two, they both are going to benefit. So a good example would be E. coli living in our intestine, a non-toxin producing E. coli living in our intestine. They're blocking attachment sites for possible pathogens. They are using nutrients or producing acids that can inhibit potential pathogens. Some bacteria even make vitamins for us. Isn't that cool? In commensalism, one partner benefits, and it's kind of neutral for the other partner. The other partner is neither harm nor do they benefit. So a lot of us carry Staphylococcus aureus in our nose, probably 10, 15, maybe 20% of us. And in that case, a Staphylococcus aureus, if it's not invading the tissues, it would be considered a commensal. It's benefiting because it has a warm, moist, nutrient-rich place to live, on us. And as long as they're just staying on the surface of the skin, they're not harming us, nor are we being benefited. So we could say that's a commensal. And then this is where we get into infectious disease. So parasitism is when one partner, and we'll call that the pathogen or the parasite benefits, but at the cost to the other partner, which we'll call the host. So any of the microbial pathogens we discussed would be an example of parasitism. So this is a classic one. So Mycobacterium tuberculosis living in our lungs, we're definitely hurt, and the mycobacterium is benefiting because, again, we're providing a warm, moist, nutrient-rich place for them to grow. Now, really, something that's really, really important, folks, is to remember these relationships can change. And we always want to remember that microbial mutualists or commensals in us humans, sometimes their symbiotic relationship changes to parasitism, given the right opportunity. So this is not to make you worried. You don't have to memorize all of the members of the normal microbiota or microbiome. Just giving you an idea of how rich and diverse the microbes that live on and within us are. And again, normally, they're commensals or mutualists, but we do want to remember, they help us or they do no damage unless special opportunities arise. So the members of the normal microbiome or microbiota, they can become opportunistic pathogens, meaning they can switch over to being parasites if given the proper opportunity. So for example, if your patient for any reason is immunosuppressed, maybe they are undergoing cancer therapy, maybe they're receiving maybe steroids for some kind of chronic inflammatory disease. They could have an autoimmune disease and thus are on immunosuppressive drugs. Um, they could be an organ transplant recipient and thus they'd be on immunosuppressive drugs. They might have, let's say, if a patient has HIV and they progress to AIDS, then they definitely have a suppressed immune system. Factors that will change the makeup of the normal microbiota. And this is a classic one, you guys. Let's say you give a patient, maybe a female patient has a bacterial cystitis, a bacterial bladder infection. So you put the patient on broad spectrum antibiotics. It kills all the good protective members of the microbiome or microbiota, and thus opportunistic pathogens like the yeast Candida albicans now has no competition, right? So it can grow to really high numbers. You can end up then with vaginal yeast infections, in women, in everybody, men, women, children, babies. You can end up with anal rectal yeast infections, and you can also have oral pharyngeal yeast infections, so-called thrush. And then this is another example where our commensals or mutualists can switch to being parasites if they get introduced into an unusual site. So let's say the Staph aureus living in our nose, maybe we have a cut and we get the Staph aureus into the cut, right? So that's a great opportunity for the Staph aureus to start multiplying and spreading throughout the body. Any kind of wound, um, um, a puncture wound, maybe from a plant or a bite, or if we cut ourselves, or if we have surgery, those are all opportunities for microbes that normally would be living on the outside of us, on the surface of us, to get down deeper, and then they can cause harm. When we introduce artificial surfaces into our bodies, like IV catheters, urinary catheters, artificial joints, like artificial hip joints, um, um, artificial heart valves. These are all wonderful places for bacteria to form those biofilms. And normally they shouldn't be forming biofilms on normal host tissues, but they love to form biofilm on these artificial surfaces. So that's another opportunity for them to switch from being a commensal or a mutualist into being a parasite. And folks, because we're next going to launch into a discussion of reservoirs and transmission, I just wanted to give a quick little blurb here on the science of epidemiology. So literally it's a study of epidemics, outbreaks of disease. And the reason epidemiology is so important, it gives us a chance to study the reservoirs and transmission. And this is important because it lets us take steps to stop transmission. And that's so we can decrease disease incidence and prevalence. So epidemiology is a big part of public health microbiology. In public health microbiology, we're thinking of populations of humans, not just the individuals. So in public health epidemiology, again, we're trying to study the reservoirs and transmission, trying to figure out ways to stop the transmission, right? So this would be another way of perhaps rephrasing epidemiology or tracking infectious diseases, trying to understand the reservoirs and transmission. And later on, from the OpenStack microbiology book, we have some slides showing you some important historical figures that were involved in developing epidemiology. John Snow, not the Jon Snow of Game of Thrones, right? Florence Nightingale, Joseph Lister, and also one that was left out, you guys, would be Ignaz Semmelweis, and we'll see those slides in just a little bit. John Snow is actually considered the father of epidemiology, and we'll explore that in a little bit. So folks, in the first PowerPoint, where we were looking at how to be a good pathogen, remember we said most microbes aren't pathogens, so you have to have some special characteristics. So in the first PowerPoint, we were exploring virulence factors. So we were kind of, at this point, we started in the first PowerPoint saying, okay, the microbe has reached its new host, so it needs to attach to the host cells, it needs to avoid or evade the immune response. Then it's going to use the host as a source of nutrients, and in the process is going to cause harm, right? So this is where the disease comes in. And then if you're going to be a good pathogen, you have to somehow be able to leave that host, or your offspring has to leave the host and find a new host, right? So in this, in the following slides, we're going to discuss the first steps here. And the first steps in how to be a good pathogen is you have to maintain a reservoir of yourself. Think of a reservoir of water, right? Stored water. Well, if you're going to be a good pathogen, you have to have kind of a place where you're going to live, right? You can almost argue almost like a safe place that you can live and replicate. And from a human perspective, we think of three basic reservoirs: humans, animals, and the environment. And we'll try to use some examples of each of these. And then, folks, we're going to see if you're going to be a good pathogen, then you have to leave your reservoir and reach a new host. And that process is called transmission. And it can get really detailed with all the names. So I'm going to try really hard to keep it simple. So we'll be talking about three basic modes of transmission: contact transmission, vehicles, food, drugs, water, and vectors, or arthropod vectors. Okay, so that's where these next slides will be coming from. So again, folks, if you're going to be a good pathogen, you have to maintain a reservoir, which is a source of yourself, right? So we have humans, animals, and the environment. With humans, as humans as a reservoir, these are pathogens that are adapted only to survive in humans. So an example would be HIV. You're only going to, um, how should I say this? You're only going to get HIV from another human, right? So humans act as a reservoirs. The second reservoir, you guys, are animals. And again, we're thinking from a human perspective. So some microbes of animals can be transmitted to humans and then cause disease in humans. So when we have a pathogen or infectious disease in which the animals are the reservoir, we call such infectious diseases zoonoses, or what we would call the pathogen a zoonotic pathogen. And indeed, you guys, isn't it all fascinating? But the COVID-19 is definitely zoonotic. We think it either originated in a bat, or maybe was passed to these really neat animals called pangolins, and then to humans, right? So we would say that the pandemic coronavirus is zoonotic, right? It has a non-human animal reservoir. And then, folks, for pathogens that are pretty rough and tough, the environment can act as a reservoir, right? So if we have maybe really um, tough, say, fungal spores. Like think of Coccidioides immitis, right? It normally grows in the soil as a filamentous fungus, a mold, and makes its spores. And then when the soil is disturbed, then we inhale them, and then the spores germinate in us. So fungal spores often have a environment as a reservoir. And then I always think of fecal contaminated water, um, fecal contaminated soil, like in the old days when we used to use human manure is a fertilizer, and we still do today. Folks use um, animal feces is manure, right? So, and there's pathogens that can survive then in the water, the soil. So, in addition to the fungal spores, and another one, you guys, would be Clostridium. So Clostridium botulinum, Clostridium difficile, the endospores are tough and resistant, right? So the environment could act as a reservoir for them. So this is, folks, just focusing on the animal reservoirs, and again, this is kind of leading up to the pandemic coronavirus, the COVID. So um, um, pathogens which normally live in animals and then can be transferred to humans, and then the humans become infected. So we talk about the pathogen jumping hosts, right? So these pathogens would be called zoonotic pathogens. The diseases are called zoonoses. So zoonoses are infectious diseases in which the pathogen has a non-human animal reservoir. And we're going to see, folks, that bats can be really good non-human reservoirs. Rodents can be, so hantavirus, for example, these cute little um, deer mice can act as reservoirs for hantavirus. It can infect humans. Yersinia pestis, bubonic plague, and monkeypox can have prairie dogs as reservoirs. Bubonic plague, we know rodents also can act as reservoirs. Rabies, dogs, bats, skunks, opossums, foxes, right? And I put this one in parentheses, you guys, chronic wasting disease with a question mark. Remember, chronic wasting disease is the prion disease of deer, elk, and moose, and it's not documented as a zoonotic. But remember, with those doggone prions, they're really sneaky at being able to jump hosts. So remember the tragic story of the evolution of bovine spongiform encephalopathy in Britain, or mad cow disease, where bone and blood meal, which was made from the bodies of sheep and goats that have died at scrapie and had the scrapie prion in it, was added to cattle feed. And then the scrapie prion was capable of binding to the cattle prions and causing them to misfold, which led to the emergence of BSE, mad cow disease. And then, tragically, when young people ate meat from the cattle that had the BSE prion in it, the BSE prion bound to the young people's good prions and triggered them to misfold. And that led to this tragic infectious form of Creutzfeldt-Jacobs disease, the ingestive form, which we call variant Creutzfeldt-Jacobs disease. So in chronic wasting disease, also being a prion, that's why I'm like, question mark. I wonder if down the road they will discover that chronic wasting disease can also be zoonotic, meaning if we eat venison or elk or moose meat from an animal suffering with chronic wasting disease, could we then develop a new form of a new variant of Creutzfeldt-Jacobs disease in humans? So again, folks, this is a lot of text, and again, you don't have to memorize all of this. If there's specific questions I want you to know about a lot of these very texty slides or links, I'll make sure to include it in the practice quizzes so you won't be taken by surprise. But folks, bats are so important. Bats are so cool. They eat so many harmful insects, so they indirectly protect our crops. They eat lots of mosquitoes, so they're they're absolutely protecting us from a lot of mosquito-borne diseases. Some plants have to be pollinated by bats. From folk stories, bats get a lot of bad news. Like you think of Dracula, and vampire bats and all of that. So I think it's just really, really important that we protect our bats. It'd be an ecological disaster if we were to lose them, right? But one thing that is really fascinating about them is they do appear to act as a reservoir for a lot of viruses. And indeed, folks, they think that the original reservoir of the COVID-19, the pandemic coronavirus, were bats. And they think bats were also the reservoir for the SARS coronavirus, the MERS coronavirus, for Ebola. There's another viral disease called Nipah. And so folks have been fascinated. It's like, why can bats live with so many viruses? And they truly are good reservoirs, you guys, because the viruses don't hurt the bats. Usually we think of, if you're an animal reservoir, you kind of co-evolved to live with that virus so they don't cause harm. And so one of the things in this just little article, you guys, I'll just point out, so bats are the only flying mammals. Okay, and this is just talking about their good quality. They devour disease-carrying insects by the ton. They're essential in pollination of many fruits like bananas, avocados, and mangoes. And this is the concern. They're also an incredibly diverse group, making up about a quarter of all mammalian species. Sorry, and this is the main point, but their ability to coexist with viruses that can spill over to other animals, including humans, can have devastating consequences. And this can happen, you guys, in some cultures. Bats are eaten. They can come into contact with other animals and transmit the virus to other animals, and then we might eat those animals, which we think maybe is what happened with, possibly with SARS, MERS, and the pandemic coronavirus, right? And then another thing is when us humans, when we invade their homes, right? We keep invading and destroying natural habitats, and that puts us in greater contact with bats. More text here, you guys, on the other thing. So this I thought was really cool, kind of nerdy, but in trying to understand how do bats live with so many viruses without the viruses hurting them. So just this little blurb here, you guys. So let's see here. So scientists reported of how bats handle something called DNA sensing. The energy demands of flight are so great the cells in the body break down and release bits of DNA that are then floating around where they shouldn't. Mammals, including bats, have ways to identify and respond to such bits of DNA, which might indicate an invasion of a disease-causing organism, like a virus. But in bats, they found evolution has weakened that system, which would normally cause inflammation. And again, you guys are going to make a big deal about this, how when sometimes we have an exaggerated immune response, like kind of an inflammatory response that goes out of control, it's the immune response that can often end up killing us. And they think possibly that's what's going on, at least with some of the victims of COVID-19. Right? So bats, because they're always generating these bits of DNA from flight, bats have evolved, they've lost some genes involved in that response, which makes sense because the inflammation itself can be very damaging to the body. They have a weakened response, but it is still there. Thus, this weakened response may allow them to maintain a balanced state of effective response, immune response, but not over response against the viruses. And again, you guys, this again, just I know I'm making you crazy, but this is what we think happens with a lot of viral infections. The virus maybe doesn't do all the damage. It's our inappropriate, kind of overboard immune response that contributes to a lot of damage in our tissues. So, and again, you guys, I just so stress how we need to protect the bats. So the bats, they have to be studied, their physiology understood, the viruses they harbor monitored for the sake of public health. That does not mean that bats are to blame for the outbreaks. As others have pointed out, humans have encroached on the lives of bats, not vice versa. And this, you guys, is really important. And again, I'm a meat eater, and so I'm kind of a hypocrite here because just in general, if humans could reduce their meat intake, right, and in some parts of the world where they talk about food insecurity, basically people are starving. Sometimes the only meat that people can afford is like wildlife, right? So they talk about bush meat. So is humans, if we could reduce meat consumption, and the other thing is maybe try to make sure that people have enough to eat, all around the world. I know that sounds kind of naive, but again, you guys, this with the at least with the SARS coronavirus and the COVID-19, it's thought that selling wild animals, wild meat in markets, especially in the wet markets where the animals are still alive, this is probably an important link in how these bat viruses eventually get into humans, right? But this is not to how should I say this? Not to think there's no hope. Um, but this particular researcher was saying that these outbreaks are probably going to be inevitable, right? And especially as we are continuing to encroach upon wild animals' homes, right? We're coming into contact with them. So it's really, really important that we continue to study and monitor um, the health and the potential pathogens that wild animals like bats can harbor. So again, folks, this was just kind of showing this key connection. I think this is a Chinese horseshoe bat, right? And I think it's got like a I want to say like a grasshopper or something here, guys. Just again emphasizing how important they are in controlling arthropod pests of our food crops, and uh, in eating tons of mosquitoes, right? That could transfer um, viruses and malaria and filarial worms to us, right? But there's been three outbreaks of coronaviruses, three different coronaviruses that we think the bats carry. And the first one you might remember, it was what almost like 18, almost 20 years ago, when they think the original SARS coronavirus 1, severe acute respiratory syndrome, they think what might have happened is that bats infected these civet cats. And again, it was the same thing in a wet market where the civet cats were kept in cages and then people buy them and then eat them. They think it was bat to civet cat and then to humans, and that caused the SARS outbreak, again, almost 20 years ago. And then in about 10 years later, they think another coronavirus with a bat reservoir was transmitted to camels, and then the camels transmitted it to humans, and that called MERS, a Middle Eastern Respiratory Syndrome. And then the last event here at the very end of 2019, and leading, of course, into this year, is possibly the horseshoe bats transferred the COVID-19 to these cool animals. You guys, I never knew about these guys. These are actually mammals. They're called pangolins, and sadly they're eaten and they're endangered. So again, in one of the wet markets, and it is outlawed in China to sell them, so this was something that was occurring with a lot of things that's done illegally. But the pangolins are eaten, used in maybe like folk medicine. So one hypothesis is bats transferred the COVID to the pangolins, and then in the wet markets, the infected pangolins then infected humans, right? Um, it's possible that in some of these infections, the virus goes directly from the bats into humans because bats are eaten. And I've read some sources say like tissues from bats can be used in folk medicine. And I read in one place they said where bat feces might be used to treat asthma, which to me is like, that sounds very dangerous. Yeah. So again, you guys, just we with zoonotic diseases, we never want to say, oh, let's just kill the reservoir, and that will get rid of it, because like I said, it would be an ecological disaster for us to lose our bats. And we're already worried here, you guys, in the United States, because of the fungal disease called white nose syndrome that is killing a lot of our bats. And this was just an article, you guys, from Berkeley, talking about how they track through RNA sequencing the phylogeny, the evolutionary relationships of the coronaviruses that led to SARS, right? So it came as bats, infected civets, and then eventually ended up infecting humans. And the wet markets where live animals are sold, and so live animals, all of their secretions can be rich in potential zoonotic pathogens. So historically, this is how, most meat was sold. Meat and poultry was that you'd go to the market and you'd buy the animal live, and then you'd take it home and you'd butcher it yourself. One of the challenges is that often you have a lot of live animals, domesticated and wild animals, crowded together in cages. Um, I haven't seen pictures of the Wuhan wet market. This is in Hong Kong. But one of my colleagues said that some of the photos she saw was really disturbing, like animals piled in cages, one on top of one another, so they're defecating and urinating on one another. They're going to be totally stressed out. And when you have a stressed animal, that's usually when they're going to shed lots and lots of viruses. Um, so, yeah, this is a tough one, you guys. Yeah, a lot of people prefer buying animals in the wet markets because they want to make sure the animal is healthy, right, ahead of time. But gosh, this, this, since the wet markets are an important place for these potentially zoonotic viruses to jump host, this might be something that maybe we could, maybe clean them up. More space, right? More space, not have animals piled on top of one another. And definitely, if we could reduce, eliminate the selling wild animals, that would be really good ecologically, and also for human health. That would be really good. So, in addition to wet markets, there's the sale of bushmeat. And when I think of bushmeat, I don't think of the live animal. I think of the wild animal that was captured and killed, and then the meat is sold. And this is another incredible way for potentially zoonotic viruses to jump into us humans. Um, and it's of huge ecological impact because a lot of the endangered wild animals, they're being eaten, right? And folks, just to give some perspective, this is a virologist down at Stanford, um, who's done some amazing work trying to set up a um, viral network where there's stations all around the world monitoring for the presence of viruses that have made the first jump, have just jumped from an animal into a human. So he's got a great TED Talk. It's a little bit old now, but it's still really good, you guys, called Hunting Jungle Viruses. And I think he made such an important point because he said very often the hunters, right, that are hunting the wild animals and then selling it, they're often really poor, and they're simply trying, to make a living for their family, right? They're very poor, and this is one way, they can make a living. So I think before we're critical or we criticize other people, we have to really kind of walk in their shoes and look at the social economic factors that they're faced with, right? And if people are starving, right, your kids are starving, um, it makes sense to me, like, if you don't have enough money to own livestock yourself, you go hunting wild animals, right? That was, again, you guys, just that was how my mother's family was raised, right? They were pretty poor, and so they hunted deer and elk to have enough food for their family. So again, you guys, I don't want to sound like a hypocrite here, but especially with us hunting, the wild animals, right? This is probably where we're going to see most of our emerging infectious diseases in the future, because researchers do believe that most of the emerging infectious diseases in the future will be zoonoses as we encroach more and more on wild animals' habitat. Zoonoses table, you guys, so a lot of these we know already, so I'm going to just go through this very quickly. Okay, so you guys, so now we've talked about potential reservoirs, and now we're going to talk about transmission. And this is how the pathogen would leave its reservoir and find a new host. And you guys, this could be a whole talk all on its own. So, and again, what I'll try to do is on the practice quiz, kind of give you example questions. So there's three basic modes of disease transmission. So contact transmission, vehicle transmission, and vector transmission, right? And so let's back up here. So you guys in contact transmission, there's two basic types, and sometimes people put in a third type. So direct contact is when the infected host has direct physical contact with a new host, and that's when the pathogen is transferred. In indirect contact, the infected host need not be in physical contact with a new host for the pathogen to be transmitted, right? And then sometimes what's included is a third type of contact transmission, which in this table is included here in indirect contact, is what's called droplet transmission. So this is when we would sneeze or cough. Even sometimes when we're talking, it's embarrassing. I notice sometimes when I'm talking, like droplets of spit are flying out of my mouth, right? So if I had some pathogen present, those droplets would be infectious. And we'll see later, you guys, there's, I guess, a lot of controversy over it. Some people say droplet transmission is when the droplet actually lands in your eye, your mouth, your nose, and in contrast to inhaling the pathogen. I know some of this just gets nerdy, right? But we'll see with COVID-19 transmission, this becomes kind of important, right? And then in addition, you guys, in indirect contact transmission, we'll talk about fomites. And fomites are inanimate objects like drinking glasses, toothbrushes, toys that can become contaminated with a pathogen. Like punctures. Say, remember you guys the rusty nail with the Clostridium tetani endospores on it, right? So fomites are inanimate objects that become contaminated with the pathogens, and when a new host comes along, has contact with the fomite, then they get infected, right? Okay, and then in vehicle transmission, airborne. This is when the pathogen can survive for a long period of time in the air, and so the pathogen can be transferred for like more than 6 feet. This is what's so funny, you guys. So for droplet transmission, droplets are supposed to spread 3 to 6 feet or 1 to 2 meters, and then to be airborne, you're being spread further than that 6 feet, that 2 meters. Okay, so dust particles, fungal, like Coccidioides immitis spores, right? Fungal spores are great. Waterborne. So I always think of fecal contamination, you guys, for waterborne, and then foodborne. So maybe the meat itself, the meat or the poultry has the pathogen in it, or maybe it's fecal contamination. And then vector transmission, this is arthropod vector transmission. So we'll talk briefly about mechanical transmission, biological transmission. Okay. So I had an old cowboy um, um, professor. And again, you guys, I have great respect for cowboys. My uncles were cowboys. You start riding a horse before you can walk. So this professor, he had his cowboy hat and his, cool Western shirt and his big buckle and his cowboy boots, and he had kind of a drawl, and I can almost repeat it because in Colorado, my family all had this Colorado drawl, right? So it was kind of, well, I thought kind of funny, guys. So he came in and he was giving the introduction on transmission of infectious diseases. So in those old days, it was Kodachromes on a screen, and he had a pointer. And so what he would do is he'd say, now remember the F's of transmission, and he'd whap the Kodachrome, right? The F's of transmission: fingers, whack; food, whack; feces, whack; flies, whack; fleas, whack; fomites, whack; and then he would pause. And it was like, I'm very nerdy, right? So I'm trying to madly write all this down. I'm waiting for the next F, right? And I couldn't figure it out by myself, but later one of my colleagues said it was fornication. Okay, all right, you guys. So the F's of transmission, right? Fingers, flies, food, fomites. Yeah. Okay. So folks, this is a contact transmission. And again, folks, I will definitely try to have practice quiz questions on this because to me, this can get so confusing. All right, so contact transmission, all right. So in direct contact transmission, I think of you guys as physical contact with the infected host. And in this, you guys, there's two different types of direct contact. Vertical, and this I think of as mom to baby, right? And this we really worry about, you guys, because the baby's immune system is totally undeveloped. So when we have vertical transmission of a pathogen from mom to baby, this can be really devastating for the baby. And then you guys, I think of all other direct contact transmission as horizontal. Okay, so to me, I just try to remember vertical is mom to baby, and all the rest of direct contact is horizontal. And then, as we said, you guys, in indirect contact transmission, fomites is really important, right? Those inanimate objects that get contaminated with pathogens. And this is the big one, you guys, with the COVID-19 droplet transmission. So with droplet transmission, the droplet containing the pathogen that you've emitted from coughing, sneezing, the spittle that we release when we talk or sing, those droplets, if I understand it correctly, they're supposed to land on your mucous membrane. And so droplet transmission occurs when you're very close, but not in physical contact with the infected host. So you're 1 to 2 meters. So I think of a meter, you guys, as 3 feet. So 3 to 6 feet. So this is where that social distancing with COVID-19 comes from, because I think what everybody thinks is it's droplet transmission. That's what we're hoping, right? Because it would be horrible if the COVID-19 can remain infectious in the air for long periods of time and be transmitted beyond that 6 feet, right? But I think this is where the 6 feet social distancing is coming from. So again, folks, just some examples of direct contact where we're physically coming in contact with the infected host. So any kind of physical touch with the infected host, so handshaking, kissing, sex, bites, if an animal bites you, hopefully another person isn't going to bite you, right? And okay, so direct contact transmission pathogens can occur through physical contact. Many pathogens require contact with the mucous membrane to enter the body, but the host may transfer the pathogen from another point of contact, like on their hand, to a mucous membrane, the mouth or the eye, right? And I just wanted to throw in here, you guys, this concept of super spreaders, right? So a super spreader is an infected host that can spread the pathogen to many new hosts. And they've identified this, like for Ebola, they've identified it for Mycobacterium tuberculosis. They identified super spreaders with SARS back in 2002, 2003. And those super spreaders could infect like 10 other people, right? So that's an example. That's like exponential explosion of the infected people. So I'm not sure if we know that there are COVID-19 super spreaders or not. And folks, this again, to me, this is really important. This is why it's this vertical transmission. So this is again direct contact. And this is transmission from one generation mom to the next. Okay, that's kind of a vertical direction if I get my pointer to work up and down. Right, so mom to baby. And again, folks, we're so worried about this because the baby developing in utero and the neonate, the newborn baby, the immune system is not totally developed yet. So the babies are really high risk for serious damage by pathogens transmitted by the vertical route. So look, I just wanted to stress, these are pathogens which cross the placenta. There are many other pathogens that can be transmitted vertically. For example, if, say, mom has a herpes genital tract infection and she's pregnant, does a vaginal delivery. So during the vaginal delivery, the herpes can be transmitted, or chlamydia, if she's infected with chlamydia. Now here, I think this is important. They're saying that herpes can cross the placenta. And obviously, you guys, I'm not an expert on this. I thought mostly the concern was vaginal shedding during the vaginal delivery. So, but you guys, who am I? Who am I to argue with the experts? Okay, I got this out of the OpenStack microbiology book. So, sometimes having little mnemonics is helpful. So they talk about torch. for helping to remember pathogens that can cross the placenta. So torch is toxoplasma, which is a protozoal parasite. You want to have your pregnant moms not working with cat feces because cats act as a reservoir, and the working with cat feces with the toxoplasma um, in them can cause infection of the mom. Other, you guys, is syphilis, a bacterial infection caused by Treponema pallidum, chickenpox, varicella. See, some of these, I'm like, really? Chickenpox, varicella. Let me check on that one, you guys. I think, I think this was in the OpenStax microbiology book, but let me just double check on the chickenpox, varicella, you guys. Okay. Hepatitis B, okay, that does make sense. Hepatitis B, HIV, that makes sense. And fifth disease, which is a virus. Okay, so those are the others. Definitely want to check on chickenpox, varicella. Okay, and then rubella, the so-called German measles, and cytomegalovirus, which is a herpes virus, and then our human herpes virus type 1 and 2, the herpes simplex viruses, right? So again, you guys, I want to double check these on the herpes simplex viruses and the chickenpox crossing the placenta. I should have done that research before I put this together, but let me find out, you guys, okay? And so, again, we're just focusing on vertical transmission, you guys, so mom to baby. It can happen in utero with those torch pathogens that we said that can cross the placenta, or it can happen during delivery, say if mom shedding the herpes simplex viruses, or is infected with chlamydia, or infected with Neisseria gonorrhea, or infected with syphilis. And, oh, this is the heartbreak, you guys, because breast milk, they've discovered, contains infectious levels of HIV. So if mom doesn't have access to good medical care and isn't on the antiretroviral drugs, the heart therapy, and she's breastfeeding her baby, she can pass HIV to the baby, which is just horrible. So again, you guys, this is kind of repetition. I apologize. So congenital syphilis, toxoplasmosis, rubella, herpes virus, chlamydia, HIV, and hepatitis B, and just some of the devastating effects of congenital syphilis. So if mom's infected and is pregnant, then you can see infection of half to 80% of the newborns. Neonatal herpes. So 60% of women who acquire genital herpes in the third trimester of pregnancy are subclinically shedding at the time of labor. So they have to be really careful about this, you guys. So maybe mom doesn't have any clinical signs or symptoms, but she could still be shedding the herpes virus. So during a vaginal delivery, the baby can be infected, causing neonatal herpes. And again, depending, right, the mortality rates can be really high, or there can be permanent neurological damage done. And Zika virus, of course, you guys, before COVID-19, this was a huge concern. So the Zika virus it can be transmitted by mosquitoes, it can be sexually transmitted, and it can be vertically transmitted. And the bad news is, you guys, if mom gets infected with Zika while she's pregnant, the virus crosses the placenta and invades the baby, and the virus loves to grow in the baby's neurons. And so you get neurological abnormalities. In severe cases, when the baby's born, they have microcephaly, right? Because the brain hasn't grown as it should. But they found, tragically, even in babies that are born of moms who got infected with Zika while pregnant, even if the circumference of the head is okay, they can still have important neurological abnormalities later on. The Zika virus, you guys, it's transmitted by Aedes albopictus and Aedes aegypti. And previously, we did not have breeding colonies of those viruses here in California. But with global climate change, the Aedes mosquitoes are spreading. And now we do have Aedes mosquitoes here in California. And I think they've located potentially a breeding colony here in Northern California. So the Aedes mosquitoes, they transmit Zika, yellow fever, dengue, chikungunya. So this is really bad news, you guys, that we have those Aedes mosquitoes now here in California. This is just, again, you guys, just to emphasize this term fomite. You certainly want to know that for the lecture exam 3 coming up. So inanimate objects can contaminate with pathogens. So inadequately sterilized surgical or dental instruments. And HAI, you guys, one translation is healthcare associated infections. So like you go and you're not infected, You go into, the doctor, the hospital, the dentist, and you pick up an infection there. And these are often really bad infections, especially if they are bacterial. Often they tend to be multiply antibiotic resistant. This is something you guys, if you're getting tattoos or piercings, make doggone sure that the artist is using sterilized instruments, right? Dirty IV needles and syringes, when folks that are using IV drugs don't have access to clean, sterile needles and syringes, um, they're sharing needles and syringes, so they become contaminated with bloodborne pathogens like HIV, hepatitis B and C, right? So needles and syringes, incredibly powerful fomites. Also, remember, you guys, when we're talking about wound botulism, again, if folks are using literally dirty IV needles and syringes, they may be contaminated with Clostridium botulinum endospores or Clostridium tetani endospores, right? And then historically, you guys, this was so tragic. So here in the history of the United States, when the British first came, many of the indigenous people, weren't really happy with these invaders coming in. And so I think it was Amherst. I think the Amherst and Amherst College is named after. So what what I heard was that Amherst came up with this brilliant idea to take the blankets that had been used with the army smallpox victims, the soldiers that had smallpox, and they were gifted to the indigenous people, the native people of the land, purposely. And this was horrific, you guys, because smallpox is an old world disease. So through natural selection, the Europeans had some, again, through natural selection, some immunity against smallpox, but the indigenous people of the New World had no immunity against smallpox. So smallpox probably played a really important role in the so-called conquest of the New World, right? Where often it was the infectious diseases that wiped out the indigenous people, right? And so this is just a cartoon showing the smallpox outbreak amongst the indigenous people. And again, you guys, this is like this was used as a biological warfare weapon, right? And anyway, I better stop there. So fomites, again, you guys, this is from OpenStax, non-living objects that facilitate indirect transmission of pathogens, contaminated doorknobs, towels, syringes, sports equipment, mats, like if you're a wrestler, MMA, that's a big deal. And again, you guys, this is one reason with COVID, we want to keep our hands away from our face, right? Because if we come into contact with some object that is contaminated with the pandemic coronavirus, gets on our hands, and then maybe we rub our eyes or our nose, and that's how we can get infected. So keep your hands away from your face. Yeah. So you guys, and this I went overboard because I just find all of this debate fascinating. So again, going back to the indirect contact droplet transmission. So this is when an infected host is within 1 to 2 meters of you, so 3 to 6 feet, and the infected host coughs or sneezes, or when they're talking, they have little drops of spit, or if they're singing, and you're within 6 feet, then those droplets land on your mucous membranes, and then you get infected. All right, and this is a big deal because the like those of you that are going to be working in the medical settings, and if you're working with COVID-19 patients or any other patient with viral respiratory diseases, it's really important for you to know, is it droplet transmission or is it truly airborne transmission, right? So this has a lot to do with safety, and it has a lot to do right now with transmission in public spaces of COVID-19. Okay, so you guys, um, so this is from the OpenStack, and I know you guys can read, but let me just go through this really quickly. So droplet transmission, which is a form of contact, versus airborne transmission, was a form of vehicle. So short distance, long distances. The infected host is present, the infected host need not be present. So although droplet transmission over short distances is considered contact transmission, longer distance droplet transmission is considered vehicle transmission. Unlike larger particles that quickly drop out of the air, fine mucus droplets produced by coughing, sneezing can remain suspended in the air for long periods, traveling considerable distances. Droplets may desiccate means to dry out quickly to produce a droplet nucleus capable of transmitting pathogens. air temperature and humidity can have an impact on effectiveness of airborne transmission. So again, you guys, what's the importance related to COVID-19? Is it only droplet transmission, or could it also be airborne transmission? And indeed, you guys, this is kind of important. So for airborne transmission, they're saying air temperature and humidity can have an impact on the effectiveness of airborne transmission. So that relates to the big debate right now, you guys. So we know influenza transmission, when it gets hot, like in the summer, influenza transmission decreases. But then as it gets colder and moister in fall and winter, transmission increases. And so that's what they're wondering about COVID-19. If we see, as summer approaches, a decrease in transmission, what they're worried about is, will we see an increase in transmission in fall or winter, right? So we're gonna see, huh? Doggone it, I wish they could get that vaccine. And again, folks, just going a little bit overboard, so you can ignore some of this stuff, but especially those of you that are going to be working with COVID-19 patients. Okay, you remember the close direct contact within 6 feet, airborne. You can The infected host need not even be present anymore. They can come into the room and cough and sneeze and leave the room, and then you walk in, right? And if it's airborne transmission, then you could become infected. And then fomites, right? This is why we want to always keep our hands away from our face. So an infected person contaminates a surface, and then later this person could leave, and then later new host comes in and contacts that same surface with their hands, and then puts their hands up to their face, and that's how they get infected. So you guys, this is going on for so long, but you guys, this I thought was a really neat article. And this was back during the one of the Ebola outbreaks in Africa, and there was a huge debate on Ebola transmission. Was Ebola just quote unquote droplet transmission, or was it truly airborne? And so this was a great article talking about the history of it. Yeah, who is the guy? This is a guy back at Harvard, a hotshot engineer. He was one that figured out that UV could kill bacteria, and then he really got fascinated with transmission of pathogens. And so he was the one that really came up with this original model of the two different ways, um, um, the two different ways that pathogens could be transmitted by droplet or airborne, right? So large droplets fall to the ground because of gravity, just like rain. Small drops start evaporating. As they fall, eventually they become so light that they float in the air, right? So he said, it appears therefore that transmission of infection through air may take one of two forms depending upon the size of the infected droplet. So he was the one that gave the direct contact droplet route, and then the vehicle transmission airborne route. Yeah. So there's a lot of confusion between these two, and you guys, I constantly still get confused with these. So it seems like a lot of us get confused. Okay. So again, folks, with the larger viruses, and again, remember this was about Ebola, the larger droplets fall to the ground. So it's thought that Ebola is just droplet transmission. It's not airborne. But then other pathogens, right, they form those little infectious droplet nuclei, and they can spread. This would be about 1 meter. Here's 2 meters. You guys, here's our social distance here. But some of the pathogens then become those little tiny droplet nuclei, those infectious droplet nuclei, and they can remain suspended in the air for long periods of time, right, and travel far distances. So examples are TB, measles. Measles is really infectious. Chickenpox. Droplet is Ebola. So again, folks, you know, I think what we're being told is that COVID is droplet transmission, right? And that's why the 6 feet. But the concern would be, what if, in some cases, COVID can become airborne? That would be such bad news. All right, so you guys, I'm gonna, I won't read all of this because I know this is probably overkill for a lot of you. But to me, it's like really important understanding the COVID-19 transmission. Okay, and this was just a respiratory specialist talking about the CDC's recommendations, because CDC for the public seems to be saying, oh, if you stay 6 feet away from one another, you're fine. But then in hospital settings, the CDC is maybe being a little bit more protective. How should I say it? Maybe the concern is there could be airborne transmission, especially in hospitals, in closed spaces, and potentially lots and lots of people that are infected with COVID. Anyway, lots of things that we still need to figure out with COVID. So folks, so here's vehicles. So food, water, drugs. Drugs can get contaminated, right, in airborne transmission. And food, one of my favorite things, right? Food is an important vehicle of transmission for pathogens, especially of the gastrointestinal and upper respiratory tracts. Notice the glass shield above the food tray is designed to prevent pathogens injected in coughs and sneezes from entering the food. It would be fascinating, wouldn't it, you guys, for us to do some sampling here. It always, oh, I have this weirdest sense of humor. If I go to the store and there's one of these little buffets set out, right? And there's a hospital nearby one of the stores. So very often I see people coming in from the hospital in their scrubs. Maybe they have their stethoscopes, around their necks, and they come in and they get their food from these little buffets, and I'm like, you're still in your scrubs. You have your stethoscope. That could be contaminated. You could be contaminating the food here. And indeed, you guys, this was many years ago. One of the students did. She worked for the public health department, and so she would go in and sample foods from buffets. And she says, "I will never eat at a buffet again." This is horrible. I shouldn't be saying this, but you guys just be aware. The spoons, the forks, whatever, are getting contaminated. When people, if you watch, and people reach in far here, see how close their clothes come to the food, right? And so so even though I still eat at these buffets, you guys, probably at some levels, from a public health point of view, this probably isn't the best way to do it. Anyway, but I will still go to the buffet once COVID is over, and I'll still get my salads there. So anyway, something maybe not to think too much about. So water fecal contamination, waterborne transmission. So this is devastating, guys, especially for little kids that don't have access to clean drinking water. So 50,000 deaths each year from water contamination worldwide. Vector transmission, you guys. A mechanical vector is an arthropod vector that maybe walks on feces, like say filth flies walk on the feces. They get the fecal pathogens on their little feeties, and then they fly over and walk on your salad or your hamburger. So they're just transferring the pathogen. The pathogen isn't undergoing any development, any replication. And then the mechanical vector, again, they've transferred the pathogen to your food, and then you eat it, and that's how you get infected. Um, Oh gosh, you guys, and this was so fascinating. I wish we were face to face, because one semester some fruit flies from the cell and molecular fruit fly genetics got into our lab, and somehow they got they got into an agar plate, and it was fascinating because um, we incubated the plate, and you could see where the fruit fly had walked, because there were microbes growing wherever the fruit fly had walked. So that was an example of a mechanical vector. And then the other thing, you guys, was, this is horrible, but was this last year? A cockroach emerged out of one of the gas outlets. Can you imagine that? And so we put the cockroach on an agar plate, let it walk around, and then we released the cockroach outside and we incubated the plate. And again, you guys, it was fascinating. Everywhere the cockroach's body touched the agar or his feet touched the agar, there were many microbial colonies. So again, cockroaches can act as mechanical vectors, especially with fecal contaminated. Fecal pathogens, right? And then, folks, what we talked about mostly in lab were what are called biological vectors, and a biological vector is an arthropod in which the pathogen either undergoes development or replication. Okay, and usually in biological vectors, it's essential that the pathogen be allowed to develop or replicate within the biological vector. It's important in their natural history. And you guys, we won't. You just had this on your lab exam too, so I won't go over this anymore. But just recognizing arthropod vectors are really important. And again, with global climate change, you guys, the range of a lot of these vectors is expanding. So we might end up experiencing new arthropod-borne diseases here in Northern California that we'd never seen before. And this is just an example, you guys, again, from OpenStack. So this is the Aedes aegypti, the yellow fever mosquito. This is Aedes albopictus, the Asian tiger mosquito, both very aggressive biters, right? And you can see here in California, you guys, that the range of the Aedes, it's moving. Here's San Francisco Bay, so we're now getting our own breeding colonies up here in Northern California. And again, they're the vectors of yellow fever, Zika, dengue, chikungunya. So those might be new emerging diseases here in Northern California. And this, again, folks, just kind of looking at the role of humans, infectious disease transmission in a borderless world. So I took it right off of this publication, you guys. So this was an infectious disease movement in a borderless world. So they use movement and I use transmission. So these are the air routes, right? So just within 12 hours, right? They were talking about you could have a person infected with Ebola, and in 12 hours that person could leave, their home and arrive in New York City. So these human behaviors, right? Rapid transit, this really helps pathogens move around the world. And this was from an article on the challenges of globalization. And again, folks, this was from 2000 to 2003, how rapidly the SARS coronavirus spread. And again, air transport, jet transport was crucial in that. Healthcare associated infections, you guys, I think I'll stop here. And I think we just have a little bit more to go, but I'm noticing I'm starting to get hoarse. So let us stop here. It's slide 48, and let's see, we don't have that many more to go, do we? Okay, let's see. Come here. Okay. No, we're going to finish, you guys, because I think it makes more sense. We'll just go ahead and finish. So HAIs, you guys, in the old days, we would have called these hospital hospital-acquired infections or nosocomial infections, right? This is an old term, but so you would go into a doctor's office or a hospital or a dental office without an infection, and while at that health care associated um, um, place, there'd be some procedure or some exposure and you got infected there. So these are often bad news. And the reason is sometimes you're really sick, like if you're in the hospital, you're already sick, and then you get another infection on top of it all. If we're talking about bacteria, if you acquire a hospital-acquired bacterial pathogen, often they're resistant to multiple antibiotics, right? So nosocomial infection, here's another Venn diagram, you guys. So if we look at the hospital, right? So a lot of people are sick. So there's a lot of potential pathogens in the environment, right? The patients are often sick themselves, so they might just have had surgery, right? So they've cut the skin or cut the mucous membranes, or maybe they have cancer, or they're a transplant recipient, or maybe they have HIV/AIDS. So you have immunocompromised patients, and then the transmission, right? So transmission of the pathogens from infected patients to staff. The staff then can transfer to another patient. It might be that staff members, because they're immunocompetent, maybe they have potential pathogen that they're carrying as a commensal, right? But then let's say, like for me, if I was working on the burn unit, right, and I have Staph aureus in my nose, the Staph aureus isn't hurting me, but what if I um, um, shed it in the presence of one of the burn patients and they got infected? That could be devastating. And again, you guys, part of epidemiology and trying to interrupt disease transmission is, remember, Ignaz Semmelweis, who was the one that had the nerve to suggest that doctors and medical students should wash their hands after performing autopsies and before um, examining their patients. Remember, hand washing can save lives, and it's probably the simplest and maybe most effective way that we can save lives in our hospital and healthcare settings. And again, folks, this was just giving, this is from CDC, Centers for Disease Control, healthcare associated infections and antibiotic resistance. So this gives you some of the classic central line associated bloodstream infections. 1 in 6 were caused by serious antibiotic resistant pathogens. Catheter associated urinary tract infections, 1 out of 10 were caused by serious antibiotic resistant bacteria. Surgical site infections, 1 out of 7 had serious antibiotic resistance. 9 and 10 with C. diff. So with C. diff infection, Clostridium difficile cause pseudomembranous colitis. Usually a risk factor is treatment with broad spectrum antibiotics that kills all the good competing commensal bacteria. The endospores aren't destroyed, right? And then furthermore, from the feces, from the diarrhea, that's going to be chock full of endospores. So you're going to have contamination of the room wherever the patient is, right? And remember, you guys, if your hands get contaminated with those endospores, and all you're using is an alcohol hand sanitizers. Alcohol hand sanitizers won't destroy the endospores, so you could transfer the endospores to a new patient. This is one reason, you guys, why they're using the UV robots, because with long exposures to the short wavelength UV, like around 260 nanometers, long exposures, like maybe for an hour, you can inactivate endospores on surfaces, right? So that's why those UV robots are used in patients' rooms when nobody's present, right? Because remember, you guys, the short wavelength UV will cause eye damage and skin damage and can increase our risk for cancers. So you guys, really quick, the history of epidemiology. So John Snow was considered the father of epidemiology. He was a doctor during the reign of Queen Victoria, I think it was, and was a contemporary of Florence Nightingale. So a lot of stuff was going on with regard to medicine. And he's famous, you guys, because he stopped a cholera outbreak in London. And kind of the punchline here is, how did he do it? He removed the handle of the Broad Street pump. So so just some background, you guys. Vibrio cholera is a bacterium that causes cholera. So it's fecal oral transmission. You have this profuse watery diarrhea. You can rapidly dehydrate and you can die from it. Um, and so it's kind of, from the pathogen's point of view, to cause your host to have a watery diarrhea is wonderful, because all of this diarrhea, all of that is contaminated with Vibrio cholera. So it's easy for it to contaminate drinking water or food. Now, back in the day in London, especially in the poor parts of London, to get um, clean drinking water probably wasn't going to happen, especially in the poor parts of London, right? And also, especially for the poor folks, they didn't have indoor plumbing. So if you lived in a neighborhood, there'd be a pump, a neighborhood pump with a handle on it, right? So you'd bring your buckets down maybe in the morning and pump your water and take it back up to your home. Well, what was happening, and again, people at this time did not know what caused cholera, right? They did not know what caused cholera. What was happening was feces from the cholera patients, sometimes it would just get thrown out on the street, right? Again, no toilets, right? So you take your chamber pot filled with your feces from the night before and you throw it out onto the street. That would be scary walking there. Well, the feces was able to contaminate the water that was being pumped out of this Broad Street pump. So all the people that were using this Broad Street pump, their water was contaminated with Vibrio cholera. And again, you guys, nobody knew what was going on. So what Snow did, because he didn't know what was happening, he took a map of that neighborhood and he mapped where the cases were. And this is really too small to see, but these little dark squares represent cases. And so this is a classic kind of mapping in epidemiology. So he mapped where all the cases. He said there seems to be a big cluster of cases right here. So what's common? What do these people in this neighborhood that's hit so hard with cholera, what do they have in common? And he figured out that there was the Broad Street pump, and he thought, maybe it's the water from the Broad Street pump that somehow is causing these people to get infected with cholera. So again, he didn't know it was fecal contamination. He didn't know it was a bacterium. But what he did is he had the authorities remove the handle from the Broad Street pump, and then that meant the poor people had to go to other pumps. And these other pumps, you guys, the water source wasn't contaminated with feces. So in simply removing the handle of the Broad Street pump, Snow was able to reduce the number of cholera cases, right? So this is why he's kind of given the title of the father of epidemiology, right? Fascinating. And then another really important person involved in epidemiology was Florence Nightingale. And again, I do believe she was a contemporary of John Snow. And she went to try to help the British soldiers fighting in the Crimean War, and she was absolutely horrified because when she got there, the wounded soldiers, the sick soldiers, were just living in filth. Their nutrition was horrible. The conditions of the military hospital were just absolutely filthy, and so she was really good, you guys. She tried to collect data on the number of fatalities in the soldiers month by month, from various causes. Total number of dead in a particular month is equal to the area of the wedge for that month. The colored section of the wedge represents different causes of death. Wounds were in pink, preventable infectious diseases gray, right? So this is what she saw. She goes, a lot of these men are dying from infectious diseases that we could prevent, just by cleaning up the place and making sure they have good food and all others brown. So Florence Nightingale, she's kind of the heroic nurse, right? But again, you guys, she uses statistics, not well, she used numbers, right? She gathered data, right? So this is what epidemiologists do. They try to gather data and then create a model to explain why the disease is being transferred, right? And my understanding is she brought about so many changes in like hygiene and nutrition that the death rate went down significantly. This is Joseph Lister, and Joseph Lister was a tough Scottish physician, and he was really concerned with how many patients would come into the hospital and then die from infections. So he had read the work of Ignaz Semmelweis saying how the med students and doctors should wash their hands before examining their patients, and Joseph totally agreed with um, Ignaz Semmelweis, and Lister had also written of Louis Pasteur's work on the germ theory of disease. So what Lister would do is he developed this machine that would spray carbolic acid, basically a disinfectant, over surgical sites, right? And that would reduce infection during surgery. He would soak bandages in this disinfectant to wrap wounds, and again, that would decrease the wounds becoming infected. Um, and even though his colleagues, laughed at him, what happened, it soon became apparent and the public became aware, was that Lister's patients weren't dying like the other patients were, right? So, and again, he had to be tough and strong because he received a lot of ridicule. But again, folks, I should have a picture of Ignaz Semmelweis in here, because Ignaz Semmelweis was the one that really promoted that doctors and medical students should wash their hands in disinfectant after doing autopsies and before examining their patients. And he was humiliated. He had a nervous breakdown and died, which is just so sad. But his words lived on, and eventually he was vindicated by the work of Lister and Pasteur. So, folks, and this would actually be a great way to kind of end the course. We have one more unit to do on immunology, but again, folks, we want to remember that we share our world, right? And if we're talking about infectious diseases, remember it's not just it's just not humans, it's our animal colleagues here on Earth, it's our environment that we need to take care of, right? And the more damage we cause to our environment, the more we encroach on wild spaces, the more of these emerging infectious diseases we're going to be seeing. So EIDs, you guys, emerging infectious diseases, so-called new diseases within maybe like the last 30 or 40 years. So mad cow disease, avian influenza, H5N1, Ebola. And again, folks, it's predicted that most of the new emerging infectious diseases of humans in the future will be zoonoses, and these are all zoonoses, right? And these are just some examples, folks. So Ebola was zoonotic. I put bubonic plague in here. Bubonic plague is not an emerging infectious disease, but it is zoonotic. And Professor Holland gave me this cartoon, and this is why I put it here. So here's old experienced bubonic plague, right? And he's talking to new little baby Ebola, and he's saying, you have much to learn, grasshopper, right? So we can learn a lot from history, you guys. Looking at these ancient infectious diseases can give us some idea of what might happen with these new emerging infectious diseases. And over here, folks, just more zoonoses. So CAFOs, these concentrated animal feeding operations. Um, this is just a wonderful place for new emerging infectious diseases to occur when we cram. And again, you guys, I'm a meat eater, right? So right. So I'm eating meat from animals probably raised in these CAFOs, and the problem is they're crowded together, they're stressed, they're often living in really filthy conditions. What I do try to do now, you guys, if whenever I can, when I buy meat, I try to buy farm raised meat, like individual farmers, and it's very, very expensive for individual farmers to raise their livestock how they're meant to be raised, like on pasture and what have you. But one philosophy is maybe decrease the amount of meat you eat, and then try to pay more to support the farmers that are doing like grass fed and maybe free range animals. if we can't be outright vegetarian. So that's the route I'm trying to take. A lot of times these animals are living in filthy conditions, so they're given lots of antibiotics to prevent infections, and we know that's going to select for antibiotic-resistant bacteria. And there's even concern that the antibiotic-resistant bacteria shed in their feces. Often the feces is used as fertilizer, and so there's concern that the fertilizer can contaminate the fields, sometimes the pasture or hay, for example, that these animals then will eat again. So it's just yeah, yay. Bird flu. This was one reason in the wet market photos I had a live chicken there, because it was thought the highly pathogenic avian influenza H5N1. It made its jump from birds into humans in one of those live markets. And then this again, you guys, just my kind of twisted sense of humor. So here's a little pig said mad cow, and here's a cow saying swine flu. Okay. I know it's not funny, you guys. So anyway, so infectious disease of both animals and humans. In zoonoses, the pathogens transferred from animals to humans. So again, EIDs, you guys, that are zoonotic: Ebola, avian and swine influenza, man-cow disease, Lyme disease, antibiotic-resistant bacteria. The Lyme disease back east, the reservoir are deer, and here in California, it's small rodents, ground animals, ground mammals. And again, folks, just want to hit this, this one last time, the One Health Triad, right? For any of us to be healthy, we have to take care of our environment. We have to take care of our animals, right? And take care of one another. Okay, thanks for hanging in there, you guys. That was a long one. Okay, the last unit, folks, is going to be on immunology. There'll be 2 PowerPoints. One will be on nonspecific innate defenses, and then the second one will be on specific acquired adaptive immunity. And it'll for sure, the specific acquired adaptive immunity will be very superficial. I'm always so happy that you guys are going to get awesome immunology in your A&P course, right? So ours will be just kind of the appetizer for you. Okay, folks, so let's see. We're going to exit here, and then